Hi everyone, my name is Pooja. I would like to share with you our study of the size-dependent transport of bacteria through microscale structures. Solid structure with microscale geometries are widespread in many natural and human environment of microorganisms. These structure includes solid surface with their geometries modified by grown biofilm, composed of neighboring cell and extracellular polymer substance. Beside this plane surface, these microstructures are also present in our clinical environment, for instance, through biofouling surface in human bodies. On the other hand, certain geometries of microstructures, such as a protic scale on shark skin, were found to be antifouling, which impede the settlement of microorganisms. However, the interplay between these geometries of these protic structure and that of swimming microorganisms is unclear. Here, we look we look into the interplay of the geometry between the structure and the bacteria. As has been demonstrated, bacteria that propel like a pusher are intrinsically attracted to solid surface through hydrodynamic interaction. It is thus expected that geometries in such surface are potentially guide the transport of these swimmers. Bef- so, before any of such application, the role of individual cell geometries in these cell structure interaction need to be understood. To understand such size-dependent effect on transport, we introduce bacteria E. coli of varying length of array of micropillars, fabricated by PDMS through soft photolithography. The pillars are on the scale lattice, typically 30 of microns with 10 micron as a gap between these adjacent pillars. To focus on mechanical effect, we adopt the non-tumbling mutant to avoid potential active response of bacteria to their contact with surface. To investigate the long-term effect while maintaining the high spatial and temporal resolution for investigating size-dependent phenomena, we employ the tracking microscope which allow us to follow the individual bacteria as it navigates through a pillar array. By stitching all video frames, we can show how individual bacteria moves over a microscopic distance and millimeter scale. I should note that to avoid mistracking of the cell due to the complicated background image, the stage was controlled manually in 3D through customized software. Here, you can see the trajectory of bacteria with different length, and all trajectories are color coded by the cell size. Blue for the short cell and red for the longer cell. At the first glance, the short cell more often orbit around a single pillar and was thus effectively trapped, where the relativity the longer cell or this reddish one were more often found to skip through the pillar and thus effectively escape from the pillar array. Here you can see the zoom in view of for the short cell and for the long cell. And this video shows an example for the shorter cell is trapped to the pillar surface, which is orbit around the pillar for a notably longer duration before escaping and approaching the next attractive pillar. We attribute such trap state to the hydrodynamic attraction from the pillar surface. To further quantify this distinct trapping and escaping effect, we compute the probability distribution of bacteria in space shown in single unit cell with lattice size A. They appear to a critical cell length, surprisingly 7 micron for 30 micron pillar lattice. Below this critical length, as shown in my left, the bacteria are distributed near to the pillar surface and consistent with the hydrodynamic effect of a single pillar. Above this critical length, the bacteria are uniformly distributed in between the gap of this pillar and which cannot be understood simply by the hydrodynamic effect. Therefore, to understand the escaping effect for longer cell, we introduce this simple geometrical model where the bacterium is considered as a pusher of length LP, and based on the constraint from the geometry, a pusher with its size shorter than the maximum LP can potentially circulate around this single pillar without its interference from the neighboring pillar, which is shown by this yellow color. In addition to the physical length constraint from the neighboring pillar, different sign of orientation of the cell relative to this pillar can also lead to torque along the opposite direction, and that leads to distant attractive and the repulsive zone. For more detail of this geometry, you can find on archive, which is preprint. As a result, we found alternative attractive and repulsive zone around the pillar circumferences due to these geometrical constraints from its neighboring pillar. The attractive or residing zone is computed to be 1.1 radian in arc angle where the pusher will be attracted to the pillar. In the pulsive or escaping zone, which is shown by this red color, experience a repulsion. And we argue that this repulsion is more effective for the longer pusher since they are more likely to experience this neighboring pillar effect. And the maximum length of pusher restricted by geometry is also shown by this dashed line. Further, we measured this residing angle experimentally, corresponding the arc angle that bacteria stay near to the pillar surface. The, dis- the distribution of this residing peak at 1 radian, which is consistent with our model prediction. We also measure such residing angle for different lattice of pillar and which also show the similar trend as given by this model prediction, which is shown by this solid line. This nice agreement suggests the power of geometry in this pillar effect. To further employ the model for understanding the size-dependent transport, we embedded this distant attractive and repulsive zone in an agent-Bose model. In this model, bacteria follow the pillar surface robustly within the attractive zone, which is shown by this thicker black line, and and while having an escaping probability when arriving at this repulsive zone, which is shown by this thinner line. The simulated trajectories of bacteria agree nicely with the experimental observation, with a shorter cell mm, with the shorter cell size match with the simulated of escaping probability, low escaping probability, and vice versa. This size dependent escaping is consistent with the geometrical constraint and we show earlier. To further quantify the transport in pillar array, we compute the mean square displacement. To show the geometrical effect, we plotted the mean square displacement as a function of arc length, and both are normalized by the lattice size A. We showed that such plot for various escaping probability. And for all these plots, we found that crossover length delta S demarcating the ballistic and the diffusive regime. This crossover length increase with the increasing probability, which is escaping probability. We found the similar trend in our experiment with the crossover length now increase with the increasing cell size. Here you can see. And so in other words, we can say the bacteria transform more effectively with the longer sizes, which is quite controversial but agree with our geometrical intuition. Further, you can see here the maximize the prediction power of our geometrical model. We map the cell length to the escaping probability by fitting a length-dependent escaping probability. Here, all cell length are normalized by the maximum position length restricted by the geometry. The best fitted curve is given by the best matching result between the experiment and the theoretical prediction on the length-dependent this residing angle, which is the experiment shown by this dot and the theoretical shown by this dashed line. Using this length-dependent escaping probability, we also recovered that experimental measurement of this crossover length, which increased dramatically over this critical length, which we found that L over LP is 0.3. With this, in conclusion, we demonstrate the capability of probing the size-dependent transport within a single bacterial colony using the tracking microscope. We also showed that geometry may play essential role in such swimming through structure environment which lead to the enormous size dependency in diffusivity. Here, the longer cell more easily navigate through the structure obstacle. This one. And we can also say that this size dependency also indicate a potential adaptation of the morphology of the swimming microorganism to its living habitat, which is based on the mechanical interaction. With that, uh, I want to thank my lab, which is Dr. Bilnio's lab. I also want to thank Dr. Gopinath's group, which is our collaboration group, And I also want to thank you for listening my talk. With that, I'm ready to take questions. Thank you.